Hi all, welcome back to this. This is the post-lunch session. So hopefully the conference lunch hasn't made you too tired and sleepy, but virtual conference actually maybe not really. We're very pleased to have with us two speakers today. The first will be Prof. Thulani Makhalanyane from University of Pretoria, and his topic will be using genome-resolved metagenomics to understand microbial contributions to ecosystem functioning. It does sound very interesting, although it's not my field. I'm glad to learn a bit more, as many people I'm sure also are. Please go ahead, Thulani. Thank you.
Thanks, Prof. Anton. I should be sharing my screen. Please confirm if you can see it. All right. So thanks a lot for the introduction. And I want to thank Werner from the CHPC for the introduction. So we rely quite extensively for our work, and without the CHPC, it just wouldn't be possible to do most of the work that we do. So in the next 20 minutes, I will give an introduction to our research group and tell you a little bit about some of the questions that motivate the research that we do. So I'll try, given that this is a very general audience, not to get too technical, but perhaps I'll give you enough detail to give you the impression that what we do is interesting and possibly might want to collaborate at some stage. So this is our group at the University of Pretoria. So I lead a group with Dr. Oliver Bezate, who's a senior research fellow. We've got about 16 postgraduate students, all the way from honors level to PhD level. And we undertake research on a variety of different topics, and I'll take you through some of those. But maybe before we go too far, I should start by giving you exactly a definition of what we mean by microbiome. So the word microbiome essentially defines the combined genetic material of microorganisms. So that's bacteria, archaea, fungi, viruses that are present within a particular environment. So the definition also encompasses or includes the potential functional roles that is done or undertaken by these microorganisms. So of course, microorganisms are present in a range of different ecosystems. So we host microbial communities, all the way to soils and marine environments. And our research group looks at microorganisms across all of those different systems. So this very colorful image shows you one of the current understanding of the diversity of microbial life. So at the bottom, as shown in that blue arrow, is where we stand in the tree of life, with plants, our relatives, being close to us. What you will see from this tree is that eukaryotic organisms, where we belong, comprise a very small proportion of the organisms that are present across ecosystems. The majority of this tree is actually microbial, and most of it that's shown represented in this tree is bacteria. So we know, of course, over the past 2 years, just how important things that we cannot see are in regulating all aspects of our life. We've got some understanding of roughly less than 1% of the microorganisms that are out there, and the vast majority remain uncharacterized, and we know essentially nothing about their roles across different ecosystems. So our group has done some work looking at microbes from a range of different ecosystems. We've looked, for example, at how microbial communities in cold Antarctic environments are able to possess the genetic machinery to be able to survive under those cold environments. we've looked at understanding how the food web structures of microbial communities in extreme deserts actually are shaped. And we've asked other questions, for example, genes that are quite pervasive, microorganisms that are able to confer resistance to antibiotics. We've looked at how present they may be in systems like Antarctic ecosystems, but also in closer to home, in individuals that are in urban and rural locations, and we've looked at their gut microbiomes. So essentially, our work has two major questions: to understand the role, the biochemical role of microbial communities, and to understand as well, other factors such as how the effect of climate change may impact on microbial communities, and in turn, what that impact has as a feedback that may further exacerbate, for instance, the effects of climate change. So our group has about 4 different thematic areas. So we're interested in looking at microbial processes, adaptations, and potentially bioprospecting for new enzymes. At the very end of this talk, I'll give you one example at how we're using CHP resources to be able to do some of this work. We're interested in looking at the microbial ecology of local soils, so local and extreme environments, and also look at the microbial ecology that is sometimes associated with particular hosts. Host here can be anything from a plant to an animal. We're also interested in looking at the microbiomes in what I'll define as geographically strategic areas. So let's say an oil company is interested in doing some kind of bioprospecting, would be interested in looking at microbes in that environment because they would be able to give us a quicker idea of whether the ecosystem becomes disturbed and whether the ecosystem is returning to its natural state because of the rapid nature of microbial metabolisms and how quickly they're able to sense any type of ecosystem change. We also have work where we've looked at human gut microbiomes. And in this work, we've done things like understanding the effect of geographic location and how it structures the gut microbiomes that are inside us. And that has a wealth of consequences. For example, depending on the type of bacteria you have in your gut, it may determine your propensity to become obese, or whether, for example, you're able to break down certain compounds. So the current projects in our group, we've got about 5 major projects that we rely on CHP resources for. We've got projects that are looking at the microbial ecology of South African soils. So some of the questions we ask here are questions related to how, for example, antibiotic resistance genes differ between agricultural soils and how different types of agriculture affects the composition of antibiotic resistance. We've got research that's looking at human gut microbiomes, like I said earlier, and we've got 3 major programs that are ongoing. The first is funded through the National Research Foundation. In this project, we're interested in understanding the ecology, evolution, and function of marine microbiomes. And this is work that's done in the Southern Ocean. I'll talk a little bit about that. And we've got another project where we are looking at microbiomes as part of a consortium with partners in the EU. And we're also interested, as part of our ongoing studies, in understanding how permanently frozen soils, what type of microbes they harbor, and potentially the type of viruses that may be stored within these soils. This has got implications, for instance, as the Earth continues to warm, though some of those dead and buried viruses may then become accessible. So we're interested in doing some kind of bioprospecting now to help us predict the type of shifts that may occur linked to climatic shifts. So I've spoken a bit about polar and marine microbiomes. we're quite interested in our group in understanding microbes in polar and marine ecosystems. We know, of course, that South Africa has about over 3,000 kilometers of coastline. We're surrounded by both the Indian, Atlantic Ocean, and to the very south of us, the Southern Ocean, which is a very important marine ecosystem that essentially controls the world's climate. So we've got a series of projects that aim to use molecular ecology to understand these systems, to look at the microbes they harbor, and essentially the ecosystem services that we can derive from these. Part of this research may also include, for instance, bioprospecting for certain anti-cancer compounds. So there's also a strong biotechnological push within some of this research. So of course, some of you might know that South Africa has a base. So this base allows us to take part in annual voyages and cruises that are supported by the Department of Environmental Affairs, allowing us to undertake some of our research. But there's also a very strong scientific justification. I talked about the role of the Southern Ocean and how it's important for actually regulating things like heat fluxes and nutrients, and how it's important for regulating the Earth's climate. So we also know that the majority of atmospheric CO2 is actually directly controlled by the Southern Ocean through both the solubility pump and also through the biological pump. We know a lot because of the work that's done by oceanographers or physical oceanographers regarding the models that they've developed and some of the work that's done over the past 20 years in terms of the solubility pump. But we lack comparative insights regarding exactly what the microbes are doing in this environment. And our work aims to fill some of this knowledge deficit. So we want to know what microbes are out there, try and understand their exact functional contributions to ecosystem services, and come up with a better interpretation of how the effects of climate change could impact on the ecosystem services that we derive from these microorganisms. One thing that, one thing that most people may not know is that microbes are important for, things like cycling carbon in the oceans. They're important for nitrogen fixation. So if there is any kind of shift within the diversity and function of these microorganisms, it will ultimately affect the rate to which the Southern Ocean may be able to carry out its ecosystem services. So we are trying to start at the very base by understanding which microbes are out there, how they vary along the water column. So looking at microbes that are on the aphotic zones or euphotic zones, and how they vary in terms of nutrient availability and the factors that structure them. So there's quite a lot as well of support in terms of government support towards supporting fundamental research in Antarctic systems. As South Africa, we've played a role in the Scientific Committee for Antarctic Research since its very inception in 1958, and we continue to play a strong oversight role. And our work in the Southern Ocean contributes to some of the mandate or activities of the Scientific OFSCA. There's also ongoing efforts and discussions to establish a sort of global microbiome initiative. And we've been in discussions with the Department of Science and Innovation and others on how South Africa may play a strong leading role in part of these efforts. Because not only because it's important to know what microbes are out there and the effects of environmental change, but also by understanding these microbes, we can use part of their functional attributes, so the enzymes that they possess, to do some biotechnological innovations. And this, of course, has strong IP implications in terms of the country and potentially products that may be directly attributable from this biodiversity. So maybe here, I'll give you just a quick overview of some of the computational methods that we use in the CHPC. So I know that there's a bunch of people with different interests, but what I wanted to show here is a lot of what we do after collecting the samples from the particular environment and processing them in the lab and getting next-gen sequencing actually depends on us being able to run computational analysis. So what I've mentioned a few slides earlier is that microbes are quite ubiquitous across a range of ecosystems, right? So a gram of soil can contain up to billions of microorganisms. So you can imagine that such big data requires strong computational resources to be able to get to a point where you know the type of microorganisms that are there and exactly what they do. So what we essentially get, right, are the raw sequencing reads. We do some quality processing steps, we assemble the genomes, and this allows us to get an idea of the diversity of microorganisms that are out there, and also by doing some annotations to get some insight of the functional roles of these microbes. So the methods that we use have advanced so much that essentially from extracting DNA from a particular soil or a water sample, without knowing what microbes are there, as classical microbiologists, people would look at microbes by isolating in the lab and looking at them under the microscope. We're able to use computational tools to start accessing the vast majority of organisms that cannot be grown in the lab. So these methods have advanced to such an extent that we can also reconstruct the entire microbial genomes of these organisms. So giving us an idea of who they are, and essentially also look at their functional genes to understand the biological processes these organisms are able to mediate. So also, there's a range of different pipelines that we use. One of the ones that we use on the CHPC servers is a program called Atlas. This allows us to carry out the bioinformatic analysis that ultimately get us to the point where we can study the diversity of organisms that are out there. And also, we can do things like try and look at potential viruses that may be linked to particular microorganisms. Right. So that's just a quick overview. So in the next 5 minutes or so, I'll give you 3 examples of where we've implemented these studies. The first example is a study that we did in Limpopo that was carried out by one of the honor students in our group. So in this project, we wanted to see whether we could get to a point of developing potentially new methods of determining the levels of contamination in water. So we got a bunch of samples from pristine sites that are not contaminated, and also some samples that were subject to human waste contamination and also agricultural waste contamination. So what Nyasha did, she used the methods that I outlined earlier to reconstruct different bacterial genomes in these communities. And what you can see on your left is that these water systems actually harbor a wide diversity of microorganisms. The tests that we typically conduct, the fecal coliform counts, do not give us a good approximation of all the organisms that may be contained in this water. By also looking at that sequence data, she was able to use these to probe what are known as Crest Phage sequences. These are viruses that are specifically linked to human waste. And what you can see from this study and what's shown in peach are the samples that we retrieved from our study. So the work that she did substantially expands the diversity of Crest phages that are actually known and described in literature. But her work, more importantly, shows that the current tests that are used to assess water quality are actually very insufficient, and we require new ways to determine other potential contaminants that may be present in water sources. Running quickly out of time, I will tell you one other story. So this is a project that was done by a PhD student, Percy, who got some samples from the South Atlantic under the auspices of cruises that are done using the SA apparatus. So Percy's questions were specifically to understand how viruses essentially control bacteria and impact on the ecosystem services that bacteria are able to provide. So the plots that are shown in the two plots on the right actually show you how diverse the microbial assemblages are in the systems that we study. You would expect that marine waters actually don't harbor as many bacteria, but this is not what emerged when we looked at some of Percy's data. Much more importantly, Percy was able to use bioinformatic approaches to actually show that these viruses, through what are called auxiliary metabolic genes, are actually able to control some of their hosts and play a role specifically in mediating phosphorus acquisition and phosphorus cycling in marine environments. Right? So, of course, you'll remember at the start, I talked about how what's been known has been mostly work that has been done through physical oceanography, but his work actually shows that microbial communities, bacteria and archaea, play a substantial role in mediating ecosystem services that occur in the ocean. So the last or the penultimate story, as I have a few minutes left, revolves around work that's been done by Diego. So Diego was actually able to join part of the seasonal experiments called SCALE that were supported by the National Research Foundation. So one of the most interesting things, without talking in detail about his work, is that Diego was able to find a substantial number of potentially novel genomes. About 200 different bacteria are potentially species that have not been reported previously. And this demonstrates how the Southern Ocean, because of its unique environmental conditions, has essentially some microbial endemism in terms of the taxa that are present within this environment. There's a lot more to say about this study, but I just simply do not have the time because I want to share one more work. And this is work that's ongoing, that's linked to some work that's done by Oliver. And in this work, we are trying to apply machine learning approaches to go back and do some bioprospecting from the various sequence data that we've acquired over the years. So specifically in this work, and it's also an ongoing study, we've applied various machine learning approaches to bioprospect for biosynthetic gene clusters. In this plot, which is quite busy, what you will see in the different colors and also shown in this plot is that there's a high degree of biosynthetic gene clusters that are already reported. What's indicated in the arrows here are some of the sequences that are uniquely from the samples that we've acquired from various biomes in South Africa. And what this plot really essentially tells you is the high degree of potential novelty an untapped biotechnological resource that we have in soils and other environments within South Africa. And this could be, a potential area which could start up a lot of different potential businesses in terms of some of the products that we can derive from our unique ecosystems. So with that, and having gone about 2 minutes over time, I'll stop here and thank you for listening.
Oh, this is pretty good. Thank you so much, Thulani. We welcome questions. In fact, you can put in questions at any time during the talk. In fact, there can be a slight delay. I'm trying to check if we have any questions yet. No, I don't. So as I wait to see if any questions arrive, just yet there. So I'm wondering a couple of things from a CHPC perspective. With your calculations and the simulations, are they memory or CPU intensive?
So it depends on exactly the application that we are doing. But yes, some of them can be quite intensive in both terms. So how we do our work is that we've got a local server where we can, for instance, start applying some of the algorithms that we use. But essentially, when we need to scale up, so run more samples, would usually scale up and use CHP resources. So there are some restrictions, for instance, like the 72-hour limits that make it a bit difficult because some of the processes and the size of the data are so vast that they essentially require a lot more time in terms of the computational resources.
Okay, yeah, like often with the calculations on the biological side, the checkpointing of your result of the calculation is not really possible. I don't know with the codes you do. Many other non-biological codes do allow checkpointing, which means you can stop the calculation. Wherever the calculation stops a short time before that, you've got a file checkpoint where you can continue the calculation from there.
So in the flow diagram I showed, essentially, we're not running a single point analysis. There's various things that we do. So things that range all the way from doing a quality control of the data to see what type of data you have. And each of those are discrete or different programs that you're actually running in the entire workflow. So in all of them, of course, there is a checkpoint, as you term, where you can essentially stop and assess the quality of the data, maybe export some plots and view in R or other software to see exactly what you're working with before then continuing with the rest of the analysis.
Oh, that is good. So then the 70 to the 96 hours or 48 hours is not that so problematic, and you probably have worked out how many cores and nodes are optimal for the calculation. So okay, I can see there are a couple of questions here now. So Quinn says, thanks Thulani, great presentation. Are your HPC workflows mostly compute bound, communication bound, memory bound, or other? It seems to be similar to what I asked, but he's also asking a little bit more than me.
Yeah, exactly. I mean, maybe to answer it, it depends on, again, to go back to the workflow, on exactly what we're doing, right? Because and some things in terms of looking at, the quality of the data may not be as memory intensive, but other processes like, generating the genome assemblies, those do require quite a high degree of computational resources, and they also take a long time. So depending also on the size of the input data set that you're using.
Okay, Quinn has asked another question regarding the Southern Ocean microbiome diversity. Are the novel species related to any particular biological functionality or action?
Yeah, so what if I understand the question correctly, so we've done some searches to look at the type of functional genes that these microbes contain. So they have a vast suite of functional processes that are linked to processes linked to carbon, nitrogen, and phosphorus acquisition. And all these genes are present. But what was quite interesting in the work that we did through the CHPC is that often most of these analyses are restricted to microbes that are on essentially the surface. And we essentially have a black box of what happens in the deeper levels of the ocean. And our work shows that there's interesting mechanisms that rely on using inorganic carbon in order to carry out functional processes. So these microbes are quite, that also is linked to the high degree of very unique organisms. So it actually goes against some of the oceanographic studies where people believe that nothing happens at the depth of the ocean. But there is some evidence from our study that there's actually potentially quite a lot of activity which occurs there. And this has implications, in terms of calculating things like the carbon budget and so on. So
yes, I remember seeing the documentary David Attenborough, and it's amazing how much life there is under there in the Southern Ocean, under under the ice and how deep it is. It's like crawling with stuff. They're not very fast moving always, but it's so that's the so you can't really access too much information on those organisms, but that's maybe sometime there will be. I don't know, we have only got about a minute to go, so I think maybe we should switch over and wait a bit, unless you've got another quick comment, Thulani.
No, no other comments, but I want to say thanks again, to everyone at the CHPC. I know sometimes things are difficult, load shedding, but I mean, we really couldn't do most of what we do without your support.
Okay, sorry about all of that, but we're trying, and sorry about all the problems that we have. Thank you, Thulani, and we'll wait another minute or so we can see that we can start with Catherine's talk, so that we have enough for people moving between sessions. Thanks again, Thulani. Where is this one? Okay, proceed now. Okay, let me just get this in order. Lost the file. There we go. Okay, right. So, where is that anyway? Sorry about this. Is messed up. Let me just. Welcome to the second half of the session, after lunch session. Please note that you can put in your questions earlier, even before the speaker has finished the talk, and there can be a slight delay in the questions coming through, so that would be better if possible. We welcome Catharine Esterhuysen, who I've actually known for years. I remember from the 90s, we used to interact as students at the Indaba conferences. And Catharine is from the University of Stellenbosch, and she's talking about computational chemistry methods in understanding the properties of porous materials. And please go ahead, Catharine. Thank you. It will also be very interesting.
Thank you so much, Anton. Let me quickly share my screen. Let me know if you can see it. Okay. All right. I hope that everything is okay. So thank you very much for the opportunity to tell you about the work that we've been doing at the CHP on non-covalent interactions, understanding those in the context of porous compounds. So what is a porous compound? Well, a porous compound is a compound that has spaces inside the substance. So we're looking at solid substances that have spaces, and those spaces can then be filled with other molecules. What we're going to talk about are various things in terms of the interactions, and we're going to talk about a host, which is the porous compound itself, and then the guests, which are substances that move into and out of that host framework. And we're particularly interested in looking at gases, and the color coding here that you should be interested in is this one over here, which is, oh, sorry, I've forgotten to talk about the other interactions that we're going to be talking about, are guest-guest, and then also host-host interactions that may be of interest. So this molecule over here that we're interested in is carbon dioxide. And carbon dioxide obviously is interesting because of the fact, well, you've just heard from Thulani's work, that it's a greenhouse gas. And we are very interested in finding porous compounds that we can use to, first of all, separate out carbon dioxide from mixtures of gases, and then also sequester them. So in other words, store them inside these porous compounds. And then in future, and I have a student who's just started doing some work in that, then actually look at doing chemistry inside these porous compounds so that we can then change this carbon dioxide to something else that will be more useful, and in so doing, remove carbon dioxide from the environment. So how does it work? So carbon dioxide has two different aspects that is useful in terms of getting it to sit inside a porous compound. One of these is shown by these double bonds here, which means that we have a pi system. So there are electrons that are moving around this carbon dioxide that allow them to overlap and form strong interactions with the framework material. The other thing that is useful is the fact that the carbon dioxide has a charge distribution. So the carbon itself is positively charged, and the oxygens, being electronegative, are negatively charged. And that charge profile means that it can interact with the host framework in specific ways. So for instance, if we have a look at, so these are three different frameworks, and I'll show you what the frameworks look like in a moment, and they're different sizes, which is, so this is not just the same thing 3 times, it's 3 different frameworks which have 3 different channel sizes. And what I've done here is I've colored them according to the charge distribution. So blue is negatively charged, red is positively charged, and green is sort of slightly negatively charged. And what we're looking for in terms of this charge distribution relative to the framework is where there is an opposite charge distribution. So we want a positive, so a red part to interact with this part of the carbon dioxide, and a blue part to interact with this part of the carbon dioxide. And what we've done here is we've actually mapped this the charge distribution given by an electrostatic surface potential of the framework onto the carbon dioxide. And we've done this using the Material Studio program under the auspices of the CHPC, and particularly looked at the VAMP module. And what this tells us is that if we compare the complementary charge distribution of the framework to that on the carbon dioxide, that this framework, so this is the second framework, has the best charge distribution, and it means that the interactions between the carbon dioxide will be the strongest for this particular framework. What does this mean for us? Well, here are the frameworks. This is what they look like, and you can see that they're very similar. They just have this different ligand, a linker over here, which makes that pore size slightly larger, and the pore is given by the sort of yellow transparent surface. And what we would expect from a larger pore size is that the carbon dioxide would get lost more easily from this framework than from this one. And this is exactly what we see if we just heat this up. If we heat up the framework with the carbon dioxide in it, the carbon dioxide comes out very easily from the first one. So this is just showing how the weight is lost as a result of the carbon dioxide coming out, and then it's much less or slower for the second one, and slowest for the third one. However, that charge distribution means that if we just have look at changing pressures, which is what we would do if we were trying to get carbon dioxide to be stored in a framework. That second framework, despite the fact that it is bigger than the first one, actually holds onto the carbon dioxide much tighter. And we can see that using this absorption isotherm, where the uptake is less for number 2 because it's smaller, but it holds on to the carbon dioxide much more tightly as the pressure is dropped. So that means that if we pressurize the framework with a high amount of pressure, we can then release that pressure, and it will still the framework will still hold onto the pressure, hold onto the carbon dioxide, but a fairly low pressure, which means that we can store it fairly safely with just a low back pressure onto the framework. So this is for a framework that is called a metal organic framework, and this is rigid. So that channel that you see there stays constant. But ideally, what we want to have is a material that starts off small when there's no carbon dioxide in there, that it kind of pops open when we have the carbon dioxide in the framework, so that it's easy to transport. Then before we have the carbon dioxide in there, put the carbon dioxide in there, and then the carbon dioxide is held quite tightly. So what we can do is actually change this from being a rigid framework to being a flexible framework. And this is an example of such a flexible framework. And you can see that this is carbon dioxide, and this is a very different behavior to what we see for methane and for nitrogen. And we have to pressurize it up fairly high, but at around about 35 bars worth of carbon dioxide pressure, the whole system pops open and absorbs the carbon dioxide. And the carbon dioxide, as we then slowly release the pressure, that carbon dioxide is retained in the framework at fairly low pressures, although we see that there's the step here where some of the carbon dioxide actually comes out of the system again before the whole thing collapses down at around about 15 bars of pressure. What's happening here? So again, this is an experimental result, but something that we can explain using the facilities at the CHPC. And we used two different software programs for studying this. We used CP2K, which is looking at periodic systems. And we also used a Material Studio again, the CASTEP and Zorption modules. And what we were able to do with this is to establish where the carbon dioxide is actually sitting. And this is one of the problems with experimental work, and why it's absolutely vital that we use computational data to study these systems, is that it's very difficult to get an experimental picture of what's happening with carbon dioxide inside a pore. We were not able to do this experimentally. We just know what happened to the framework, but we don't know what happened to the carbon dioxide. And so the computational results help us to identify what's happening with the carbon dioxide in the framework. And this is at the high pressure form, where we have two molecules of carbon dioxide in each one of these pores. And we can see now that those carbon dioxide molecules are stabilized inside that framework by interactions between these carboxylate groups. There's a CC bond over here, which is also stabilizing the carbon dioxide. And we also see that there are guest-guest interactions, so interactions between the carbon dioxides, which are actually stabilizing the whole thing. And from the computational results, we can actually compare the electron densities in the frameworks and with and without the carbon dioxide, so that we can see what is driving the stabilization of the carbon dioxide within the framework, and why does it work to hold the carbon dioxide inside the pores. And we can see that the carbon dioxide, as the pressure is released, the carbon dioxide moves position very slightly, the structure changes slightly, but the interactions continue on, and that's why the carbon dioxide can remain inside the pore at a really fairly low pressure of overpressure of carbon dioxide. So this is very useful for understanding why carbon dioxide sits in a framework like this, but it's a very fundamental kind of picture. And what we're interested in doing is looking at how we can actually use this kind of information practically to design new, better materials that are able to separate carbon dioxide and then store it. We have the computational work that I'm describing here, and we also do a lot of experimental work. So I have a colleague, Len Barber, who is a guru at designing new instrumentation. And one of the things that he has done is to look at, to design a very sort of small scale instrument that will help us to test materials for pressure swing adsorption instruments, which would be what we would need to use in terms of separating carbon dioxide from the atmosphere. So we don't have a carbon dioxide, nitrogen. We don't have an overall air testing kit at the moment. What we have is a way of mixing carbon dioxide and nitrogen to give us an idea of how we can actually just separate at the moment those two specific components, with the idea that eventually we will move towards separating carbon dioxide from air. So in other words, we need to look at using this information that we have, for instance, here, that nitrogen is not easily taken up into a framework, whereas carbon dioxide is taken up into a framework, in order to be able to try and separate nitrogen from carbon dioxide. So you would think that this would be fairly simple to do. So for instance, this is zeolite 13X, which is a zeolite that is being used for carbon dioxide separation. And you can see here that if we look at experimental sorption isotherms, that the uptake of nitrogen by zeolite 13X is much, much less than by carbon dioxide by zeolite 13X. So the idea being that we should be able to take this information, and if we just have zeolite 13X and we have a mixture of carbon dioxide and nitrogen, that we would basically separate out carbon dioxide, and that would be easy. Unfortunately, it's not so simple, and we see this even when we do simulations. So for instance, this is a simulation using the sorption module of Materials Studio, and you can see that it's giving us some false information. It looks like it should be fairly easy to separate out the carbon dioxide and the nitrogen, but we can see that there is a small amount of uptake of nitrogen, and this is not modeled currently within the setup of the sorption module as we had it here. So we needed to work out what the issues were. Now, this is not just a computational problem. This is a general issue within the separations sort of community, because to get measurements of the separation of carbon dioxide and nitrogen is really difficult. So this is actually why we've designed and built an instrument that will allow us to look at sorption of mixtures of carbon dioxide, oxide and nitrogen, because it's hard to do experimentally. So what people tend to do is they take this information here for single gases, and they combine it using the ideal adsorbed solution theory, which is then used to predict what the uptake of, say, carbon dioxide and nitrogen is by some substance, in this case, zeolite 13X, at different pressures. And you can see now, just now we had two-dimensional picture of pressure versus uptake. Now we need a second dimension here. We need to look at the composition. So this is what the theory tells us, that as the pressure increases, the uptake of carbon dioxide should increase dramatically, and nitrogen, there's not going to be a heck of a lot of uptake. However, when we measure this experimentally, you can see that carbon dioxide is fairly well described, but the nitrogen is not well described at all. There is much greater uptake of nitrogen at high pressures and at high compositions of nitrogen than is predicted by the ideal adsorbed solution theory process. Now, what this means is that if we tried to use zeolite 13X to separate nitrogen and carbon dioxide, we get a bad separation if we're looking at pressures that are relevant for pressure swing adsorption instrumentation. So, for instance, which is around about 3 bars, and you can see at 3 bars at 80% composition of nitrogen, the uptake is significantly higher than we would want, and there would be quite a lot of contamination of the carbon dioxide with nitrogen as a result. So we need to understand how this works in order to be able to get a better picture of what's going on. What's interesting is that this is not true for all substances. So for instance, this is a similar kind of picture for a different compound called copper H-cust1, which is a metal organic framework. And this is the CO2 and nitrogen uptake. And you can see that the uptake increases as the pressure increases and the composition of CO2, or in this case N2, increases. But here, these little numbers that you can see are actually the deviations between what the ideal adsorbed solution theory tells us and what we get experimentally. And you can see that there are very, very tiny deviations here. So for this particular compound, the description is really quite good. And we actually wanted to understand why it is that zeolite 13X is really badly explained by that theory, and why this one is not, or explains it very well, because one of the things that we want to do is go and test a lot of different compounds and try and identify what compounds give us the best separation. So we need to understand how these models can be used for people who do not have access to instrumentation like we do. So we started off with modeling these using Material Studio, the CASTEP and Absorption modules, and we realized that the issue with the picture that I showed you a few slides ago of the two absorption isotherms, where the nitrogen is completely underpredicted, had to do with the charges. And the absorption module is a module that is based on classical principles, and the charge distribution was wrong. So we used CASTEP to help us get two better charge distributions. So CASTEP is ab initio based, and we used the Hirschfeld and Mulliken charges derived from CASTEP, and then brought those into the absorption module and got a better picture of what is happening within the absorption process. This helped us to work out which charge distribution we needed to use. We then once we had a good methodology that we knew would give us results that compared to the experimental results, we could then start looking at explaining the results in terms of what's actually going on inside the pores. And we looked at different pressures. So again, this is still the CASTEP and absorption modules within Material Studio. And we noticed that the difference between the behavior of the carbon dioxide and the nitrogen is that when nitrogen is in 100% or is present 100%, well, not in a mixture, that the interaction energy that it undergoes with the framework is very different to what happens when it is with carbon dioxide. So in other words, the nitrogen interacts with the carbon dioxide with a stronger interaction than it has with the nitrogen. it's or with itself or with the framework. And we can see this very clearly by plotting the interaction energies for 100% CO2 and 100% N2 versus those the interaction energies that occur when we have mixtures of carbon dioxide and nitrogen. And you can see that there's very little change between the interaction energy of the carbon dioxide in the framework, whether there's nitrogen or whether there is not. But for the nitrogen, that carbon dioxide in there makes a huge difference on how stabilized nitrogen is within the framework. So the carbon dioxide actually helps to stabilize the nitrogen in the framework. Copper H-cast, on the other hand, looks somewhat different, and you can see that there's very little change between what happens when we have 100% CO2 or 100% nitrogen and when we have mixtures. And this explains why we see a much more a much more ideal kind of uptake with the copper H-cast one than with the zeolite. This has to do with the where it is being taken up, and you can see for zeolite, the carbon dioxide mainly sits here, whereas the nitrogen is kind of spread over the whole framework. Whereas for copper H-cast 1, there's very clear distribution, difference in distribution. Carbon dioxide sits here and the nitrogen sits here. So they don't interfere with each other. And this has to do with how they are absorbed into the zeolite. And Anton is looking at me, so I'm guessing I'm running out of time. So I'm going to quickly skip through this and just say that we are able to identify positions within the framework where the carbon dioxide and the nitrogen sit, which allow us to explain why we have this difference between ideal behavior with the copper H-cast and the non-ideal behavior with the zeolite 13X. So in summary, I hope that I've been able to show you that computational chemistry enables us to get a fundamental understanding of the role of intermolecular interactions on the properties of porous materials. We're able to do this on the basis of strength of interactions, which we would not be able to interpret any other way than if we had computational results. It gives us information about sorption sites, which is usually very difficult to determine experimentally. We can even look at competition in mixed gas systems, which again is very difficult to do experimentally. And this helps us to understand why we get ideal versus non-ideal sorption. And the application for this is that we will then be able to design materials and processes that will enable us to separate carbon dioxide and then to store it safely. So finally, I just want to thank a few people, the students who were involved in this course, in this project. Without them, obviously nothing would have happened, my collaborators, but mainly, of course, thank you to the CHPC Anton Lopez. Thank you so much for all your hard work. We bother you on a regular basis, the same with Krishna, and you are always willing to help out, and we are so grateful for that. So thank you very much, and thank you to everyone for your attention.
Thank you, Catherine. It was very interesting to see how the work is progressing after each time I see one of your talks.
Yeah, something new every time.
Yep. I don't see any questions as yet.
I've scared everyone.
I think you may have answered some of the things I was wondering about. The interactions between different molecules within different unit cells. How much is that accounted for within the calculations, and how significant are those? And
oh yeah, so they're very important. So what we do is we do a periodic. This is one of the reasons we use Material Studio, CASTEP, CP2K, is that they're all periodic system, or they can describe the periodicity. So it's not just within unit cells; it's then between unit cells and so on. They all play a role in stabilization, and it's different things in different compounds that can result in stabilization.
If you have an empty cavity in some neighboring ones, you looked at those kind of things. Yes. Not all of them we filled in reality. So
Yes, exactly. So one of the reasons, for instance, that copper H-cust is the carbon dioxide and the nitrogen don't interfere with each other is because the carbon dioxide sits in one type of pore and the nitrogen sits in a different type of pore, and then they don't interfere with each other. They don't interact and so on. So that's why you have this stabilization of the two different sorts and why it behaves ideally.
I see a question from Quinn now. Thank you, Catherine. This kind of research is critical as we push forward with carbon mitigation. Have you done any work looking at selectively removing other related molecules, for example, CO, out of COH2 mixtures?
So not yet. This is one of the things that we would very much like to look at. We're definitely moving in that direction. Now that we kind of we've developed methodologies that work, and we can explain a lot of these things on a fundamental level. We can now take that and apply that to other systems, to other guests, and so on. But yes, definitely that's something that we want to get to.
Okay, and I was wondering, you did refer to some parameterization that you've done. The models in VAMP, have you had to adjust any of the other potentials or any other
So yeah, it's sometimes this can be quite tricky because, for instance, so that one I think wasn't too difficult because I think it was zinc, but sometimes if we're looking at metal organic frameworks that have different metal centers, Compass does it. So we often use Compass as a force field, and that has some of the metals, but not all of the metals that we work with. So then things become a little bit complicated, and sometimes we need to parameterize the metal centers to get a better description, a successful description of the interactions.
Okay. No. Quinn said it will have a huge industrial relevance. Thanks.
Absolutely, absolutely. So at the moment we've been kind of doing fundamental stuff just so that we understand what's going on, but now we want to move into actually solving industrial problems, because yeah, I mean it's almost too late, right? So we really need to do something now.
Sure. I think that's all we have time for the moment. Thank you so much, and good to hear how I progress in each time.
Thanks very much.
Thank you, Thulani, as well as Catherine. Thank you.